we're going to talk about how the metabolism of bacteria can change depending on their growth phase. So when you get some bacteria and you start growing them in fresh medium, they go through 4 growth phases. First of all, they go through lag phase, which is basically when they're still getting used to the new medium and they're not growing yet. Then they go into phase 2, exponential phase. This is the phase in which the bacteria are growing and dividing and eating all the food that they find inside the growth medium. But then they can run out of food. And this is when they reach phase 3, in which there's an equilibrium between the growth and division of the bacteria and the death of bacteria. And finally, when there's not enough food left in the medium, the bacteria start dying and you're in phase 4, which is death phase. These researchers were interested in finding out what happens to the bacteria's metabolism in these different phases. Specifically, what happens to the metabolism between exponential phase or growth phase and stationary phase, which is the equilibrium phase that we have just seen. To study this, they worked with marine bacteria and they focused on the proteins inside of these bacteria. In order to study the proteins, these researchers used a technique called comparative proteomics, which basically is they took bacteria that were in exponential phase and bacteria in stationary phase, and they extracted all the proteins and then analyzed them and compared them to see which proteins were present, when, and in how much quantity. So what differences did they detect in these marine bacteria's proteins in exponential phase and in stationary phase? In general, they saw there was a big difference in the proteins depending on the growth phase. Mainly, in the exponential phase, the bacteria had proteins involved in growth and division, whereas in stationary phase, the bacteria had proteins that would help them compete with other bacteria for the limited amount of nutrients that were in the environment. For example, one way to compete with other bacteria when there are limited nutrients in the environment is to insert inside the membrane a larger amount of transporters, and that way the bacteria would be able to use these transporters to uptake more amount of nutrients from the environment and that way compete in a more effective way against other bacteria that haven't got so many transporters. So the researchers saw that this is in fact what the bacteria were doing in stationary phase. And not only that, they were also devoting resources to store nutrients inside the cell. For example, the bacteria in stationary phase stored carbon in large molecules that they could then use later on when they didn't have enough food in the environment. Another interesting thing that these researchers saw were that the 3 different types of bacteria that they studied had different changes in metabolism. Each bacteria had a specific strategy for changing their growth phase from exponential to stationary phase. And these strategies that are characteristic for each bacteria can be considered signature metabolisms that are specific for each bacteria. So I want you to keep in mind that you can find out a lot about bacterial metabolism by studying the proteins inside them with techniques such as, for example, comparative proteomics. And also that bacteria are able to change their metabolism depending on what growth phase they're in, so that they can either specialize in growing and dividing or in competing with other bacteria when there are a limited amount of nutrients in the growth medium. I hope you've enjoyed this video. As always, I have left the link in the description. And if you have really enjoyed this video, you're welcome to give me a thumbs up or comment or subscribe to my channel. And I'll see you next week.